I'm very happy to be here for the first time in the function COSI. I will explain how we use local energetic frustration to understand the evolution of proteins through time and species, and perhaps to make biophysics great again, paraphrasing Raphael from last talk. So the concept that you have to have in mind first to understand my talk is that proteins are not optimized only to fold, but also to function. And these two things are often in conflict with each other. So what's frustration? To understand this concept, we can make use of the energy landscapes theory that says that proteins are very specific systems that have been selected by evolution in such a way that most of the interactions that are present in the native state are good for folding. And this is known as the minimum frustration principle. However, whenever you look at the native structure of a protein, there are always some interactions that are in conflict with the local surroundings. And we call these interactions highly frustrated. And these conflicts are there because they might have been positively selected by evolution. Otherwise, they would have been minimized energetically over time. And if we think that the native state of a protein is a collection of conformers that exist in a dynamic equilibrium, we can think of these local frustrated interactions as the ones that facilitate the motions of the protein in between these different conformers and eventually provide function. We have a tool from some years ago that is named frustatometer that allows us to localize and quantify these conflicts. So we get a protein structure and it gives you a classification of contacts and single residues in different colors that we can interpret in this way. So whatever is red is highly frustrated and likely related to function, and it means that the native energy in that loci is not optimized compared to random variants that can occur. Whatever is green is minimally frustrated, and this means that the native energy in that loci is optimized compared to random variants, and is most likely related to foldability and stability. In a data set that is representative of the PDB, it was shown that up to 10% of the interactions are highly frustrated. They are clustered in different sites of the protein. 40% of the interactions are minimally frustrated, mostly located in the hydrophobic core, and the rest are neutral. So over the years, different studies from my PhD supervisors group and with me over the years have shown that highly frustrated interactions can be linked to different functional aspects of proteins, such as binding sites, allosteric sites, and active sites in enzymes. However, all these studies have been shown that this is true, but it is true for very large and heterogeneous data sets. But what about if you want to understand local frustration in single proteins? This was still kind of difficult to interpret because it needs a lot of prior knowledge to make sense of the frustration patterns in a protein structure. And to make an example of this, I want to go back to the very beginning when the local frustration algorithm was published in 2007, when there was also an accompanying article where the authors shown which are the functional consequences of frustration in a protein called IM7. IM7 is a very short protein that exists in bacteria, and it binds another protein that's called colicin E7. So these are the frustration patterns of the protein. You can see there in the magenta circle that the binding sites, these two tyrosines, are highly frustrated. And this frustration is released after binding. But also the authors have shown that this frustration is responsible for the emergence of an on-pathway folding intermediate. And this is something that is kind of complicated because if you have folding intermediates and highly frustrated interactions that are exposed, this has been shown some years ago, this can lead to aberrant phenotypes like aggregation or misfolding. And evolution has found a lot of strategies to deal with this. For example, the invention of chaperones. Chaperones, we know also from some recent studies that they recognize non-specifically highly frustrated interactions, and then they sequester the client protein and only release it in the presence of the target protein. But again, this is a lot of prior knowledge. And I said the binding residues are highly frustrated. But what about the other residues that are in those other circles? Are they relevant? Are they important for something? Are they just there transient in some conformation, but not really relevant at all? So the question I started to have in my mind some years ago is, can we use evolution to better understand frustration? Can we use frustration also to better understand evolution? So let's suppose that we have this protein from humans. We calculate frustration, we classify the different residues in these classes, and then we transfer the colors to the sequences. And the question is again, are these colors significant? Are these colors important for our understanding of the functional consequences of this protein? So what we can do is we can take the same protein in different organisms. We calculate frustration for all of them, and we map the frustration colors to a multiple sequence alignment, and we get a multiple sequence frustration alignment. And we can see that there are certain regions where frustration is not conserved, and there are other regions where frustration is highly conserved in all the members of a family. And if everything is conserved, it might be because there's some selective pressure. So we can use information content to convert this multiple sequence frustration alignment into a set of sequence and frustration logos, where the tallness of the bars mean how conserved the identities of the residues or the frustration states of the residues are in the different columns of this multiple frustration alignment. And then we can see, for example, if this is our protein of interest, the one in the top, and you see that asparagine there that is highly frustrated, that asparagine is not highly frustrated in the entire evolutionary ensemble, whereas the tyrosine in that side is highly frustrated and conserved in most of the members of the family, and therefore it's very likely that it's functionally relevant. So we have put all of these ideas into a new tool that has been recently published that we call FrustraEvo to do this automatically for whatever set of structures you might want. And if we go back to the example of IM7 and try to understand what's relevant and what's not, we can take the sequence of IM7, blast it against the NCBI database, and we can just get, for example, the first 100 hits. We reduce the sequence redundancy, and we end up with 21 homologs. Many of these homologs do not have a structure. We predicted the structures we have for 2, and then we gave the structures and the multiple sequence alignment to FrustraEvo and see what we see. And actually, if you see, the first row is the queried protein, and we see that that one that says no important is because that frustration actually is not conserved. So we don't know what it's doing there. Might be because of conformational issues, I don't know. But the other ones, the 2 tyrosines, are highly frustrated and conserved in all the family. But something that we didn't know, and it was not reported in 2007, was this tryptophan in position 75 that is highly frustrated in most of the members of the family. We checked the literature, and we observed that this tryptophan actually is important to stabilize the folding intermediate. And the question we had when we were writing the paper is, but wait, wasn't that this intermediate in folding was something undesirable and a consequence of the binding sites that were highly frustrated? So the question was, maybe the functional intermediate is functional itself. We didn't have an answer to this question, but we wanted to show how FrustraEvo can be used to pose and to create new hypotheses about the functional consequences of a given protein. So all good so far. I explained how we can use FrustraEvo to understand a single protein family, but we wanted to go a bit farther. Can we use FrustraEvo to understand the evolution of superfamilies? So one of my favorite examples is hemoglobin. Hemoglobin is very interesting because it's composed of 2 alpha globins and 2 beta globins, and these 2 families are evolutionarily related. So whatever difference in sequence or frustration that you may see today is the consequence of their functional divergence over time since their common ancestor. So we took 21 hemoglobins from mammalians, and we repeated our analysis. You can see that in the first row, I have the results for mixing together alpha and beta globins. Then you have alpha and beta. You can see that alpha and beta have different positions that are highly frustrated, but the arrow there points at a position that is what is known as specificity defining position, which is a position that gives identity to the 2 classes, alpha and beta. And you can see that in alpha, it's this highly frustrated lysine, therefore might be functional, whereas in beta, it's not functional, or we don't know, it's a glutamine. So if we check all the positions that are tall and red, we were able to link them all to protein-protein interaction sites within alpha and beta, alpha and a chaperone that prevents it from misfolding, alpha and heme, and so on and so forth. But then, what was first, the lysine or the Q? And it was very interesting because there was a paper from some years ago where they made a phylogenetic tree and they reconstruct the ancestors of some of the internal nodes, and then we can get the sequences, predict the structures, measure frustration, and we see that the ancestor proteins actually are the ones that had the lysine that was highly frustrated. And it was later in evolution that beta globins decreased the conflicts and loci and became less frustrated. And you see, we included there neuroglobins and myoglobins that are monomeric proteins. Therefore, they completely minimize the conflicts in that because they don't need this protein-protein interaction site anymore. Therefore, they don't need this conflict. So to conclude this part is local frustration, I'm sure, that can be used to understand the evolutionary constraints in protein families, both related to stability and function. Differential frustration patterns can help us to understand the divergent evolutionary history of superfamilies. And a question I start to have is, okay, these globin families are different, and this is something, solutions that evolution have found, but what other solutions might be there? Can we go further and understand how different folds can accommodate different sequences and how vast or not this space is? So many years ago, I was really excited about this paper from Johannes Soding, it's called A Galaxy of Folds, where they compare the scope domains in sequence. And whatever is close in this plot, it means that it's similar in sequence, and it's very likely to have almost the exact same fold with different sequence signatures. So the question I started to have is, what are the biophysical limits of protein families? What are the biophysical limits of protein folds? What are the accessible sequence and structure spaces for a given fold? So to study this, Miriam Polley, a first-year PhD student from our group, she started to use protein MPNN. Protein MPNN is an algorithm from the Baker's group that can generate sequences that may fit the coordinates of a given protein structure. So this is called reverse folding, right? So what Miriam did is she took all the members from the globins and all the members from the beta-lactamases. She generated, for example, 30 sequences to fit the structures of each of the member. And then we predicted the structures of all of these sequences with ESM fold because it's faster than AlphaFold. And then she selected the one that maximizes the PDT score for each family member. And then she uses protein MPNN again to evaluate what are the biophysical properties of these ensembles in the design sequences. So these are the results. In the first part, you have the natural globins that I shown before, and you remember that the tall red bars are all or most of them involved in protein-protein interaction sites. But the sequences in the lower row that are designed by protein MPNN only recover as conserved things that are tall and green. So they only recover the positions that are important for the stability of the fold. And this is not surprising because the authors of protein MPNN from the Baker group, they say that this algorithm might generate sequences that more strongly encode the structures than the original native sequences, because evolution, in most cases, does not optimize for stability. Well, this sounds a little bit like what I was saying about frustration, right? So far so good. We were able to provide a biophysical interpretation of protein MPNN. It maximizes stability and buffers out the functional signal. So let's do it again for the lactamases, which is a family that we analyzed some years ago, where we found that the catalytic sites, because of functional reasons as well, they're highly frustrated and conserved. These are the asterisks in those. So we say, okay, let's do it again. We will find again that the catalytics are minimized, and we can just write a small note about it and see. So for our surprise, actually, in the top row, you have the natural lactamases. Again, the catalytic sites are tall and red because they are functionally constrained. But in protein MPNN, even when the algorithm wants to improve the overall stability of the fold, it cannot get rid of the conflicts. It retrieves the catalytic sites in their native identities, and they are still highly frustrated. So we started to say, why is this happening? Why is protein MPNN not able to get away from the conflict and actually retrieves it in a very conserved fashion? So we started to go around, or I started to go around about these papers from some years ago. They say that protein folds actually, they cannot be whatever we want. They are constrained by the laws of physics, and actually sequences diffuse in the space and they fall into folds. So the idea is that folds also constrain the available sequence space. So the idea that we start to have is it maybe that catalytic sites are just an unavoidable consequence of how folds are made by physics, and maybe catalysis is just an acceptation from these conflicts that pre-existed the need of catalysis. So in order to do that, we did the following. We retrained protein MPNN. We took away all the enzymes that belong to the same enzymatic classification as lactamases, and also we retrained another version without all the enzymes that are labeled as enzymes in the training set. And we repeated our analysis again. And this is still an ongoing story, but I can tell you that we did reverse folding for all of these things, and we retrieved again the catalytics. So this might be for two reasons. Either catalytic sites are very biased about a very specific set of residues, which is very likely, or this predefinition of sequences by structure is something true. We don't know what it is, but we are going to have answers soon, I hope. So to conclude, Frustration analysis can help us to interpret what machine learning methods are capturing. You can go to Miriam's talk also on Friday. She will use GPT-2 and other methods. Protein MPNN can generate minimally frustrated proteins. However, catalytic residues seem to be an exception. This could be either because of predefinition of sequences or structures or because of biases in the catalytic sites on compositions. And if this is true, we can use frustrable and protein MPNN. If we have a protein from a structure from a protein, we have no idea what it does. We can use this strategy to unveil residues that are functionally relevant. With that, just to thank all the people that made me possible to be here, especially Maria Freiberger, one of my first ever student with whom I collaborated for the last 10 years, Diego Ferreiro, my PhD supervisor, and Alfonso Valencia, of course, who's here, my current supervisor, and all my collaborators. Thank you very much.
Thank you for a very interesting talk. Questions? We got time for one, I think. No, we got a couple more. Yeah, we have it.
Do you have any? This is fascinating, by the way. I love it. I've been following this for many years, but I don't know the answer to this question.
Okay,
Yeah. Okay. Do you have any measure of how much modeling errors affect the measurement of frustration?
So that's a good question. Thanks. So we did many of these examples are actually made with experiments. Frustration, as we use it, is a coarse grain potential. So we repeated this with models. There's not much error. I mean, if your models have a good quality, like median average PLD score above 70 or 80, I think it's pretty much reliable. But of course, if you have priors to see if this is real, might help. But I think it's pretty reliable and fast.
Hello. So I have a question. Like most of the examples that you show are residuals that are more soluble, more polar. Is there like a pattern that you see? Like, what are the chemical type of the residuals that are more frustrated normally in a protein, and why? Like,
this is something we want to explore more. What I can tell you from preliminary results, I mean, we have been doing this for a couple of years. Yes, but it's a bias because you see them. And actually, for example, lysines. Many times when you have a highly frustrated conserved residue, it's a lysine or an aspartate, but there are also many lysines and aspartates that are not highly frustrated. So it's not at all, it's not a question of a type of specific residue. It varies according to the context.
okay, thanks.
Thank you.
Thanks a lot. So, no, is it working? Yes. Regarding the relationship between frustration and selection and evolutions, does frustration predict the amount of selection that you see within species, for example, in humans? So that if you have a highly frustrated or non-frustrated site, does it segregate at lower frequency in the population because it's under stronger constraints?
This is also something we are starting to do. I don't have the answer, but we think that yes, the variability that you observe in humans, for example, might be constrained by this because variants that may touch these highly frustrated conserved regions because of evolutionary reasons. If you touch them, it may lead to pathogenic phenotypes like cancer, for example. Other mutations might be neutral. So this is something I cannot really tell you what it is. We think we are going to try it. Maybe in a couple of years I give you a better answer. But yeah, thanks.